Welcome back to all. Welcome back to our official YouTube channel, SRNBGNR Government Arts and Science College, Khammam, and myself, Dr. B. Satish Kumar, Assistant Professor in Biochemistry. And in last few classes, we have been learning about the processes in molecular biology. In last few classes, you have learned about the replication process, transcription process, and post-transcriptional modifications. And in the previous video lesson, you have learned about the genetic code. Okay, so the genetic code is the first topic in Unit 2 of molecular biology. That Unit 2 is for the translation. Okay, so in the Unit 2 previous video class, we have learned about the genetic code. In that, what is the genetic code, the definition of genetic code, and we have covered that. And apart from that, the features of genetic code, like triplet codon, code is universal, code is without punctuation, non-overlapping, all these things have been covered in the previous lesson. So if you have the knowledge about the features of genetic code, so then we can easily translate a mRNA sequence into a amino acid sequence. So if you have given a specific amino acid sequence, a specific nucleotide sequence in mRNA, so then from starting from AUG, you can start reading each and every three nucleotides as one codon, and by looking at the genetic code table, we can easily assign the codons to specific amino acids, like that, like the example shown here in this table, in this slide. So in this slide we have the mRNA molecule, one mRNA molecule here, this mRNA molecule, and this mRNA molecule is being translated into a peptide. Okay, in this peptide we have the amino acids, specific amino acids. So starting from AUG, so the first three nucleotides AUG codes for methionine, and GCC, the next three nucleotides code for the alanine, GCC. So if you look at here, that GCC here, you will get GCC here, GCC codes for alanine. Okay, and after GCC you have UGG. So here UGG, here UGG, that is tryptophan. Here the tryptophan is. So like this, if you have the knowledge of features of genetic code and the genetic code table, so then you can easily translate this mRNA sequence into the amino acid sequence of the polypeptide, corresponding polypeptide chain. So you that means you can decode the nucleotide sequence into amino acid sequence. So that is what the relationship between nucleotide sequence and its corresponding amino acid sequence in protein, that is nothing but the genetic code. So you have learned about what is genetic code and how you can use the genetic code to specify amino acid sequence in a its corresponding polypeptide chain. Okay, so now in this class we will start with the deciphering of genetic code, that is elucidation. So that is nothing but decoding of genetic code. So how the scientists are able to decode the genetic code, how they are able to get the genetic code table that we have seen in the earlier slide. So you have this genetic code table. So how do we know this genetic code table? How the scientists recognized or formed this genetic code table? So that we are going to study in this video lesson. That is the the deciphering of genetic code. So in the deciphering of genetic code, three experiments are very important, and using these three experiments, the scientists are able to decode 64 codons to their corresponding amino acids. So we have 64 different codons to specify for 20 different amino acids. So now we have to know what is the amino acid coded by each and every codon among out of 64 codons. So we have to assign the specific amino acid for each and every codon. Of course, you know that 3 codons are stop codons. So we have only 61 codons that codes for 20 amino acids. Even that stop codon must also be elucidated by these experiments. So we have totally 3 different types of experiments, and these experiments enable the scientists to form the genetic code table. Okay, so the first among these 3 experiments is the use of homopolynucleotides. So this is a very basic experiment where the scientists Nirenberg and Mathai used synthetic mRNA molecule, and that mRNA molecules are having homopolymer sequence. So that means a mRNA molecule which contains only U residues, so poly U residues, so that is a poly U. So if we use that poly U sequence in a cell-free translation system, then what happens? You will always get only one codon, U codon. Okay, the translation system may start at random site, but it always encounters only one codon, that is U, because you have only one kind of nucleotide in the entire length of mRNA molecule. So that is why there is only one possibility of codons, that is U. So the scientists, when they used this poly U sequence, so that means you have this sequence U. So this is the mRNA. They used a cell-free translation system. So cell-free translation system very much, it comes very handy in the experiments like this, where you will have the complete machinery for the synthesis of protein. Okay, so except mRNA. So except mRNA, you have everything in it for the synthesis of polypeptide chain. So if we add a mRNA molecule to this cell-free translation system, so then what happens? This system uses this mRNA and synthesize the corresponding polypeptide chain based on the mRNA sequence. So that is why this is known as a cell-free translation system. So there are no cells in it. In fact, we have a solution which contains each and every essential ingredient for the translation process except the mRNA molecule. So when the scientists added poly-U sequence, synthetic poly-U sequence, they found that a polyphenylalanine a chain, a peptide which is having only phenylalanine residues. So that means U codes for phenylalanine. So in the same way, next time they used poly-C sequence. So then they get polyproline amino acid sequence. So that means CCC codes for proline. And next time they used poly-A that coded lysine. So that means AAA codes for lysine. So there is no ambiguity at all, and it is confirmed that U codes for phenylalanine, CCC codes for proline, and AAA codes for lysine. Okay, so but they cannot they are unable to use poly-G sequence and unable to elucidate the GGG codon, because that poly G sequence always forms a tetraplexus. So that means they form like secondary structure, tetraplex secondary structure, which cannot be utilized as mRNA molecule in a cell-free translation system. So if we add poly G to the cell-free translation system, so that won't act as the mRNA, so there won't be any corresponding peptide synthesis. So the GGG code is not elucidated in this experiment. So we have only 3 assignments out of 61 assignments, out of 64 in fact, out of 64. This experiment enabled us to elucidate or specify only 3 codons to their specific amino acids. So that is the use of homopolynucleotides. And the next experiment is use of trinucleotides. Okay, so here in the earlier case homopolynucleotide sequence, that is artificial mRNA molecules having one kind of nucleotides. Okay, but in the second case, they used a trinucleotide. It is not a complete mRNA molecule, neither a complete mRNA molecule or nor a full length one. It is a very short trinucleotide. It will have only 3 nucleotides. Okay, they chemically synthesized all 64 codons 64 codons as a trinucleotides. So they synthesized AUG, suppose, like, so that is a trinucleotide. So now that acts as mRNA. Okay, but that won't specify a amino acid because there are no other codons. So only one codon is there, then it specifies only one amino acid. So that is why in this experiment they used all the different types of, they artificially synthesized all the 64 types of nucleotides, codons. Okay, so to understand this experiment, you have to know some basics about the translation. So then only you can understand this experiment clearly. So during the translation process, what happens here? You will have a mRNA molecule, and you will always have the codons in mRNA molecules, of course. But here, what happens? The ribosomes binds to mRNA molecule. Okay, so here ribosome will have a small subunit and a large subunit. So this is ribosome. In case of prokaryotes, we have 70s. So this is 50s and this is 30s. Okay, so you have ribosomes. These ribosomes binds mRNA, and in that mRNA, in that ribosome, you will have some specific sites. So it binds one codon here, and now tRNA comes and binds to this ribosome, and you will have a codon anticodon interaction here. Okay, in the previous class I have explained about the general structure and features of the general features and concept of tRNA as an adapter molecule. So the codon and anticodon interaction you have seen in the previous class. So here you have we have a codon on mRNA, and the tRNA will have anticodon which recognizes the codon. Okay, so on the opposite side it will carry a specific amino acid. So here what happens in this experiment, we use a trinucleotide, that is AUG. Suppose AUG, we are using AUG. So now what happens, this small subunit and large subunit, the ribosome binds this trinucleotide. Okay, so now it acts as a codon. Now a specific tRNA comes and binds to this ribosome based on the anticodon. This anticodon will be specific for this codon, and because AUG codes for methionine, this amino acid is nothing but the tRNA specific that carries methionine. Okay, so this is the configuration. So if you have all these things in the machinery and except the mRNA, and if you add AUG, then what happens? The methionine coding tRNA comes and binds to here. Or otherwise, if you use UUU, then phenylalanine carrying tRNA comes and binds to ribosome. Okay, if you have CCC, then proline tRNA comes and binds to that ribosome. So this is what ribosome binding assay. Okay, so this is the configuration that appears during the translation process. You will have ribosome, you will have that specific trinucleotide sequence, and you will have a specific tRNA that carries the corresponding amino acid. Okay, so in that case, what happens? First, they used one specific nucleotide. Suppose a nucleotide. Okay, in this experiment, they have taken, the scientists taken 20 different test tubes. Suppose 1, 2, 3, and up to 20 different test tubes for 20 different amino acids. Okay, in each and every test tube they added trinucleotide. In this case, for example, AUG, they added AUG to this one, and all the 20 different test tubes. Okay, now you have trinucleotide, same trinucleotide in all 20 reaction mixtures. Okay, so now they added ribosomes for all the 20 tubes. So now you have nucleotides and ribosomes. Then what happens? This nucleotide, this codon or trinucleotide binds to ribosome. So now we have ribosomes bound to codon. And now the main difference comes here that for one they added each tube with one amino acid, radio labeled amino acid. So that means in first tube, suppose they added some amino acid 1, or suppose glycine, in the second one alanine, and in the third one serine, like that. Okay, up to 20, suppose proline. So this is, so each and every test tube there is AUG and ribosomes and a specific tRNA that carrying the specific amino acid, only one type of tRNA. Okay, that amino acid is also radio labeled. So now what happens? Because this is AUG and this is, suppose this is say for example it is 15th tube, and this, in this 15th tube, what happened? We used methionine. Then what happens? In the first tube, the AUG binds the ribosome, but it is unable to bind the tRNA molecule because the tRNA is specific for glycine. That tRNA is unable to bind or recognize AUG. Okay, because it is coded by only methionine. AUG always binds the tRNA which carries methionine, and the remaining won't bind to the ribosome. So now in this case, what happens? The tRNA will be in solution, not bound to ribosomes. So in the first tube you have the tRNA, amino acid carrying tRNA, which is known as a charged tRNA, charged with amino acid. The charged tRNA is free, It is not bound to ribosome in the first, second, third, like that. Every single 20 tubes except 15. In the 15, what happens? The tRNA carrying the charged tRNA is methionine tRNA, methionine tRNA. So that is why the tRNA binds to the ribosome. So that means in the 15th tube you have AUG, and this AUG binds the ribosomes, and you have tRNA also, that bound tRNA only in this case, that carries methionine, and you know that 15th tube is containing methionine because you added it. Okay, so now this is radio labeled. So now in this case they used they filtered all 20, 1 2 3 15 and up to 20. Okay, they filtered, they used filters, okay, and filter these solutions. So then what happens? Ribosomes are larger molecules, that is why ribosomes are retained by the filters. So the ribosomes will be on the top of the filters, and tRNAs passed, tRNAs freely move through the pores of the filters. Okay, so now, so you have ribosomes on the first, second, third, and all 20 filter papers will have the ribosomes. Okay, when we study, when we read the radioactivity, then what happens? You will find the radioactivity on the 15th filter paper, because this 15th filter paper is having ribosomes that is bound to the radio labeled methionine methionine tRNA. So you have tRNA on the ribosomes that is carrying radio labeled methionine. Okay, only in this case only the tRNA is bound to ribosome. So that is why you will get radio label radioactivity on the filter paper. Okay, in remaining all the cases, in remaining 19 cases, you won't get any radioactivity on the filter paper. Instead you will get the radioactivity in the solution which passed through the membrane, that filter paper. Okay, so that is why this is also known as filter binding assay. So in this case you can easily see that on the filter you have ribosome on the filter, and you have tRNA. This one is the tRNA. That tRNA is carrying the specific amino acids. Okay, so this is what the filter binding assay. So using that filter binding assay, each and every time they used a specific trinucleotide, and they run 20 test tubes, 20 tubes, and found out what is the amino acid that is specified by that particular codon or trinucleotide. So based on this, they are able to elucidate or specify 50 amino acids out of 64 codons. Okay, so they determined 50 codons out of 64 codons. So in case of the remaining 14 codons, they have some type of doubt. Okay, so that is why they confidently elucidated or determined or specified 50 codons to their corresponding amino acids. So that is why they required another specific experiment which can elucidate the entire genetic code without any doubt or any ambiguity. So you have the final experiment in the deciphering of genetic code, where the scientist Haragovind Khorana is the scientist who got Nobel Prize for this, and he used copolymers. Okay, he chemically synthesized RNA molecules with defined sequence. So that means he know what is the sequence inside the chemically synthesized mRNA molecule. So that is called a defined sequence, defined mRNA sequence. They were synthesized by means of copolymers by chemical process. So that is why then he knows the sequence, and when we observed the amino acid sequence, corresponding amino acid sequence, then he can easily specify the codons to its corresponding amino acid. So how it was possible, we are going to see now in this third experiment, where Hargobind Khorana used copolymers as artificial mRNA molecules in cell-free translation system. So in this experiment, Khorana used copolymer. So copolymers means a short stretch of mononucleotide. So suppose if we take a dinucleotide or trinucleotide, and if we polymerize these dinucleotides or trinucleotides, so then we will get copolymers. Okay, so here first of all he used in one experiment, he used AC, a dinucleotide, and they added all these dinucleotides in a sequence. Then what we will get? We get an mRNA containing AC, 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 AC. So like this. So what happens now, if we use this sequence, okay, this copolymer sequence in a cell-free translation system, what happens if the protein translation system starts reading at here, okay, at between from this C, then what happens? CAC, the first codon will be CAC and second codon will be ACA, okay, and the next codon again CAC. So that means if we use two repeat codon, okay, if we use a copolymer with two nucleotide repeats, okay, nucleotide repeats, then we will get two different codons, only two different codons that are alternative. So that will be in alternative arrangement. Okay, so from this he has able to find out that a a peptide is being synthesized when we used this two nucleotide repeat copolymer as mRNA. That peptide is having two amino acids. Okay, first one is threonine and second one is histidine. So that means these two codons, first codon is CAC and second codon is AC. Second codon is that ACA. So we know that only two codons are there in possible in the mRNA, and that peptide, corresponding peptide, is having only two amino acids, that is threonine and histidine. So that means these two codons are coding these two amino acids, so there is no confusion at all. So simply CAC may code for threonine or histidine. In the same way, ACA may code for threonine or histidine. So which codon is coding for which amino acid, we don't know in this experiment, but we can simply say these two codons are coding for these two amino acids. Okay, so earlier from the earlier experiment we have the knowledge of about 50 codons. Okay, so from that, if you are able to find out about CAC, what is the codon, what is the amino acid that CAC codes for from this experiment, from the second experiment, then you can easily assign ACA to the other amino acid. Okay, so if you know one codon, so then you can identify the remaining codon. Okay, so that means here he using the information which was which was the result of first experiment and second experiment. Okay, So that means it is not an independent experiment. So it uses the information from the earlier experiments. Okay, so from by using some information, you can he is able to find out the remaining information. So in this case, he assigned that CAC for histidine and ACA for threonine. So this is 100% confirmed sequence. Okay, so there is no ambiguity because it is a full length of mRNA molecule, a long length, and specific interaction takes place. Okay, so there is no ambiguity at all. And when he used in this way he used other two nucleotides. Okay, so out of 4 nucleotides, if we use 2 nucleotides as one set, that is doublet codon, like you will get 16 combination. So you can run 16 experiments by using 2 repeat copolymer, 2 nucleotide repeat mRNA. So from the 16 you can get 32 codons. Okay, so in that way, in one experiment he used different types of 2 nucleotide base copolymers, and then 3 nucleotide, he shifted to 3 nucleotide repeats, and then shifted to 4 nucleotide repeats also. In 2 nucleotide repeats you will get 2 codons, and 3 nucleotide repeat you will get 3 codons, and 4 nucleotide repeat you will get 4 codons. So in that way he is able to elucidate all the 64 codons, and this is the only experiment where the scientist, the Hargobind Khurana, is able to find out the stop codons also. Okay, the stop codons are also recognized by this experiment. So at the end of the experiment we have the genetic code table. So genetic code table is elucidated by these 3 experiments. First experiment is the use of homopolymers, and the second one is triplet binding assay. This is also known as filter binding assay, where around 50 codons have been coded. In the first experiment only 3 codons have been coded. But the third experiment conducted by Hargobind Khurana, who used the copolymers which are chemically synthesized. So that is why he is able to elucidate all 64 codons. So out of the 64 codons, as we all know, that 3 codons that are UAA, UAG and UG acts as stop codons. Okay, so this is what the genetic code, elucidation of genetic code. Okay, these 3 experiments finally revealed the relationship between the codons and their corresponding amino acids.